morning of January 21st, 2007, a local fisherman named Haruo Kanbe was working near Awashima Port on the shore of Suruga Bay, Shizuoka Prefecture, when he saw something in the shallows he couldn't identify. It looked like an eel. It moved like a snake. Its mouth, when it opened, showed rows of needle-sharp teeth throughout the jaw, each one shaped like a trident. He called Awashima Marine Park. Staff drove to the site and captured the animal using 2 nets. A female, 1.6 meters long, weighing approximately 7.5 kilograms. They had never seen one alive before. Almost nobody had. They placed it in a seawater pool at the park, filmed it, and waited. A park official told Reuters 2 days later that they believed moving pictures of a live specimen were extremely rare. Staff described the footage in their park blog as being of incalculable value. The shark died within hours. The species was a frilled shark, Chlamydosalacus anguineus, and what makes that morning matter is what the frilled shark actually is. An animal from a lineage so old and so little changed that encountering one is closer to paleontology than wildlife observation. Sharks have been leaving traces in these oceans for roughly 450 million years. Scales embedded in stone long before any creature had learned to walk on land. Descend through the water column and something consistent happens. The sharks get older. Not in individual years, but in evolutionary age. The shallower waters hold the younger dynasties. Descend far enough, and the animals waiting in the dark belong to lineages that were ancient when the dinosaurs were still alive. At the very bottom of that ladder swims the frilled shark, a design that got finished early and never needed revision. The deeper you go, the older they get. And at the end of that descent swims an animal that watched the dinosaurs end and barely noticed. The animal the Awashima staff filmed that January morning bore almost no resemblance to what most people picture when they hear the word shark. The body was long and sinuous, eel-like, the scientific literature consistently says, though the Latin name already tells you. Anguineus means snake-like. Maximum confirmed length is just under 2 meters, with the binomial and family Chlamydosalacidae formally established by American taxonomist Samuel Garman in 1884 from the first recognized specimens. Most individuals are smaller. The 2007 Awashima specimen at 1.6 meters was roughly the length of a door frame. The gill slits come first because they are what give this animal its name. Nearly all sharks have 5 pairs of gill slits. The frilled shark has 6. The first pair, the bottommost, connect across the underside of the throat, forming a ruffled collar of tissue that wraps around the neck. The genus name encodes this precisely. Chlamydosalacus comes from the Greek chlamydos, meaning cloak or mantle, and salakos, meaning a cartilaginous fish. The gill septa, the tissue between the openings, are enlarged and ruffled. When you see the animal in the Awashima footage, that collar is the first thing you register. Not just a gill opening, but something fitted around the throat, something decorative and strange. The mouth is terminal, positioned almost at the tip of the snout, rather than underslung beneath it as in most sharks. A 2020 developmental study found that this jaw position is a derived feature. The jaws elongate anteriorly late in embryonic development, migrating forward from the subterminal position standard among most shark embryos to achieve the adult terminal configuration. The frilled shark's most visually distinctive feature is a specialization built late in development, not an untouched blueprint carried forward from antiquity. That distinction matters for understanding what this animal's biology actually argues. The teeth explain the hunting. Both jaws carry multiple rows. Upper jaw row counts typically range from 19 to 28, lower jaw from 21 to 29, varying between individuals. Fishbase notes, with some dryness, that these teeth are sharp enough to inflict lacerations on the hands of the unwary scientist examining its mouth. Each tooth carries three main needle-sharp cusps with smaller cusplets between them, all of them sharply recurved, angled backward toward the throat. The mechanics follow from the shape. Once something enters that jaw, the backward-pointing cusps prevent exit. Pull forward, and you pull into the point. The grip is a mechanical commitment from the moment of contact. Stomach content studies of 139 specimens from Suruga Bay, published in the Japanese Journal of Ichthyology in 1990, established the diet plainly. Approximately 61% cephalopods, squid primarily, around 11% bony fish, and occasionally smaller sharks. The tooth design and the diet form a single coherent argument about what this animal has been built to do. There are two species of frilled shark. The second, Chlamydus alacus africana, found in southern African waters, was formally described by David Ebert and Leonard Compagno in 2009 in Zootaxa. It has a longer head, more widely spaced eyes, and more tooth rows per jaw than C. anguineus. For most of recorded scientific history, the genus was believed monotypic, a single species standing entirely alone. The African species ended that assumption, though both remain among the rarest large vertebrates in any ocean, known primarily from incidental capture. The frilled shark's depth range is most accurately expressed as 500 to 1,000 meters, roughly 1,600 to 3,300 feet for the typical population, with confirmed records spanning from near the surface to approximately 1,500 meters depth. The 2007 Awashima specimen was found in shallow coastal water. Animals adapted to hundreds of meters of pressure, to temperatures barely above freezing, to total darkness, don't surface into warm, lit shallows in good health. Kanbe's shark was already dying when he spotted it. To understand what the frilled shark represents, you need the full geological distance behind it. Shark-like creatures first left traces in the fossil record approximately 450 million years ago in the late Ordovician period. Those traces are scales, tiny dermal denticles embedded in rock from what is now Colorado and Siberia, characteristic enough in structure to be identified as chondrichthyan in origin, but without teeth or skeletons to tell us what the whole animal looked like. The first confirmed shark teeth appear around 400 million years ago. The first near-complete shark skeletons, Cladocelacia, preserved in the Cleveland Shale of Ohio with such extraordinary fidelity that muscle fibers remained visible in the matrix, arrive at roughly 380 million years ago. Sharks as a lineage predate trees. Land plants capable of producing structural wood first appear at roughly 385 million years ago, some 65 million years after the earliest shark-like scales. They predate the non-avian dinosaurs by more than 200 million years. 5 mass extinction events have rolled through the history of life on Earth, and sharks in some recognizable form emerged from every one. Around 420 million years ago, give or take, as best as the fossil record and molecular data can tell us, the lineage split. One branch became the elasmobranchs, sharks, rays, skates. The other became the chimeras, the holocephali, ratfish, ghost sharks, rabbitfish. Today's chimeras are immediately recognizable as something profoundly separate. Large eyes adapted to darkness, flat tooth plates for crushing rather than rows of replaceable stabbing teeth, smooth, scaleless bodies, a long tapered tail. They look assembled from mismatched parts, because they are a design that diverged from the shark template hundreds of millions of years ago, and has since followed a wholly separate path. The deepest of the surviving chimera lineages colonized ocean depths relatively recently in geological terms. A 2024 Royal Society study suggests deep-sea crown chimeras represent a post-Jurassic radiation, but the lineage itself traces its divergence from sharks back to a world with no land-dwelling vertebrates in it. Sharks continued diversifying through the Carboniferous, a period paleontologists describe as a golden age of shark diversity, with an extraordinary range of body forms and tooth architectures. The Permian extinction, at approximately 252 million years ago, devastated marine life. Sharks survived it. By the early Jurassic, approximately 195 million years ago, a specific group had emerged that has never entirely left: the Hexanchiformes. The Natural History Museum in London states this directly. Hexanchiformes is the oldest known group of modern sharks, with a fossil record extending to the early Jurassic. An articulated skeleton, Notodontoides munsteri, preserved in the Solnhofen limestones of Germany at around 150 million years ago, gives a clear anatomical picture of the order at mid-Mesozoic. Fragmentary teeth in even older deposits carry tentative Hexanchiform attributions, though those identifications remain debated among specialists. What marks every species in Hexanchiformes is one anatomical fact. Nearly every modern shark has 5 pairs of gill slits. Every member of Hexanchiformes has 6 or 7. The broad-nosed 7-gill, Notorhynchus cepedianus, has 7. The sharp-nosed 7-gill, Heptranchius perlo, has 7. The blunt-nosed 6-gill, Hexanchus griseus, has 6. The frilled shark has 6. The 6 or 7 pairs represent an ancestral condition that the rest of modern sharks lost somewhere in the past 200 million years of evolution. Hexanchiformes simply retained the original count. 2 families comprise the order: Hexanchidae, the cow and 7-gill sharks, and Chlamydoselachidae, the frilled sharks. A 2013 mitochondrial genome analysis established the frilled shark as the sister lineage to all other extant Hexanchiformes, diverging from the rest of the order at an estimated 82 million years ago. A blueprint already ancient when the first dinosaur walked, preserved intact by an ocean that never changed the question it was asking. The blunt-nosed 6-gill, Hexanchus griseus, is the most familiar member of Hexanchiformes and among the largest sharks in any deep ocean. Verified maximum lengths cluster around 4.8 to 5.5 meters, roughly 16 to 18 feet. Adults are commonly found between 500 and 1,100 meters depth, with some records approaching 2,500 meters. They carry a 6-gilled inheritance from the early Jurassic and descend into conditions that would kill most of what swims at the surface. The 6-gill represents impressive evolutionary age. The next rung represents something else entirely. The Greenland shark, Somniosus microcephalus, belongs to the sleeper shark family, Somniosidae, a separate branch of deep evolutionary time anchored in individual lifespan rather than lineage age. One specimen has been recorded by submersible at 2,200 meters, 7,200 feet, nearly a mile and a half down, at the wreck of the SS Central America. They reach over 5 meters in length and more than 1,000 kilograms at the upper end. They grow approximately 1 centimeter per year. Sexual maturity arrives at an estimated 150 years of age. In 2016, Julius Nielsen and colleagues published in Science the study establishing the Greenland shark as the longest-lived vertebrate on record. The method was radiocarbon dating of eye lens nuclei. The center of the eye lens is composed of metabolically inert crystalline proteins formed during early embryonic development, retaining the isotopic signature of atmospheric carbon at the moment of formation, like tree rings embedded in living tissue that never subsequently changes. Nielsen examined 28 female Greenland sharks ranging from 81 to 502 centimeters in total length, comparing their lens nuclei against the known radiocarbon record, including the post-World War II nuclear testing spike that propagated through marine food webs from the late 1950s onward. The minimum confirmed lifespan for the species, 272 years. The oldest individual in the sample, estimated at approximately 392 years, with confidence intervals extending past 500. A Greenland shark alive today may have been born before the Mayflower crossed the Atlantic. The bowhead whale, previously the longevity record holder among vertebrates, reaches around 211 years. A Greenland shark born around the time of the American Revolution would still be sexually immature. Their metabolic rate matches the timescale. A 2022 study measured mean field metabolic rate at approximately 21.67 milligrams of oxygen per hour per kilogram of body mass, among the lowest ever recorded for a large vertebrate predator. Early research attributed this extreme slowness purely to cold water temperature effects. Later analysis found the slow metabolism appears intrinsic, a property of the animal itself rather than solely a consequence of its thermal environment. The cold suits what the Greenland shark already was before it reached that cold water. The Greenland shark tells you what time can do to a living thing. Centuries of it, compressed into one body, still hunting. The frilled shark tells you what time chose not to do. The genus Chlamydosalacus has a documented fossil record dating to approximately 80 to 95 million years ago. Fossil teeth attributed to extinct members of the lineage, Chlamydosalacus gracilis from Angola and C. bali from Canada, appear in Campanian stage late Cretaceous deposits, alongside animals now studied in natural history museums. The fossil teeth closely resemble those of the living animal. The lineage survived the KPG boundary at 66 million years ago. Frilled shark teeth appear in the Takatika grit at that boundary layer in the Chatham Islands of New Zealand, alongside bird fossils and conifer cone fragments, testimony to a lineage that passed through the extinction event that ended the non-avian dinosaurs. The popular claim that the frilled shark has been unchanged for 80 million years is a simplification of more layered evidence. The 2020 University of Vienna developmental study demonstrated that the terminal jaw position is derived rather than primitive, evolved late in ontogeny. The lineage has evolved. What it has maintained is a recognizable body plan, a consistent tooth architecture, and a persistent ecological niche across tens of millions of years. The scientific explanation sits in two connected principles: the nature of the deep ocean and the mechanism of stabilizing selection. Below roughly 1,000 meters, the deep sea is among the most stable environments on the planet. Temperature is near constant, typically 1 to 4 degrees Celsius, year-round, wholly independent of surface seasons. Light is absent. Pressure increases predictably with depth and does not fluctuate seasonally. The primary productivity reaching depth depends entirely on what sinks from above. Marine snow, the slow rain of organic particles from productive surface layers. The prey the frilled shark hunts, cephalopods primarily, alongside bony fish, has been present in the deep ocean in broadly similar forms throughout the documented history of the lineage. The ecological niche hasn't been structurally reorganized at any point a frilled shark lineage left evidence of existing. Evolutionary biology distinguishes two fundamental types of selection. Directional selection pushes traits toward one extreme: larger size in an arms race, faster metabolism when food grows scarce, different tooth morphology in response to a shifted prey base. Stabilizing selection operates differently. It removes individuals whose traits deviate too far from an already optimized center. In a genuinely stable environment, with consistent food sources, no novel apex predators requiring physiological response, and no seasonal pressure demanding metabolic flexibility, stabilizing selection becomes the dominant force. A design that already works faces fewer disruptions demanding revision. What survives is what selection has continuously confirmed as adequate, and what gets eliminated is everything that deviated from the working center. The conversation's synthesis of deep-sea evolutionary research put it plainly. The slowing rates of evolution in deep-sea organisms are likely a result of the relatively stable environment underwater, particularly in the deep sea. The deep sea preserves what already works, reducing the selective pressures that ordinarily force morphological change. It functions as a stabilizing chamber, a place where the disruptions that drive rapid evolution elsewhere are reduced to their minimum. The frilled shark's reproductive biology is the clearest expression of this slow life framework. Aplacental viviparous. Embryos develop inside the mother, nourished entirely by yolk, with no placental connection. Litters of 2 to 10 pups, born at roughly 40 to 60 centimeters. Year-round breeding with no detectable seasonal peak in the Suruga Bay population data. And the gestation estimate. Tanaka's 1990 study examining those 264 specimens collected over 6 years, inferred from embryo size distributions and reproductive cycles a gestation duration of up to 42 months, 3.5 years. Never directly confirmed in captivity because no frilled shark has survived in human care long enough. If the estimate holds, it represents the longest known pregnancy of any vertebrate. 3.5 years to gestate a pup roughly half a meter long. The best figure science has, from one study, from one bay, from animals that died the moment they reached the surface. Population size, unknown. Maximum individual lifespan, unknown. Behavioral ecology in the wild, inferred almost entirely from body structure and stomach contents. Whether individuals move widely between ocean basins or remain geographically restricted, debated. The KPG extinction record matters precisely because of what it reveals about the ecological profile that survived it. Deep dwelling, adapted to scarcity, not dependent on surface productivity. The frilled shark was positioned to weather a mass extinction that restructured the surface world entirely. The deep ocean is a refuge from the disruptions that force everything else to change, and the frilled shark lineage has been in that refuge long enough to carry teeth through the end of the Mesozoic and out the other side. Since Samuel Garman formally described Chlamydococcus anguineus in 1884, nearly every significant encounter with a living specimen has come from something else catching one by chance. The largest systematic study ever conducted on the species, Tanaka's 1990 reproductive work, drew its 264 specimens from 6 years of targeted surgested shrimp trawling in Suruga Bay. The frilled shark enters science as bycatch in a shrimp fishery. That is the foundation of almost everything known about its reproduction. Beyond Suruga Bay, the record is a scattered case file of accidents across 140 years of global fisheries operation. Multiple specimens have been documented from trawl fisheries off the coasts of New South Wales, Tasmania, and Victoria in Australia. Portuguese trawlers working off Madeira in the Northeast Atlantic have produced records. The Northeast Atlantic population, part of a discontinuous distribution extending from Norway in the north to Namibia in the south. New Zealand waters have produced multiple records, the distribution suggesting the southwestern Pacific is a concentration area for the species. South Africa and the western North Atlantic contribute additional specimens. A paper in the Bulletin of the Museum of Comparative Zoology documents what appears to be the first in situ observation of a living frilled shark in the western North Atlantic, a submersible encounter rare enough to require its own dedicated publication. Earth Touch News notes that since the species description, frilled sharks have been documented in over 20 countries. That sounds like a broad record, and the geographic range is real. But consider what it took to assemble. 140 years of global fisheries, hundreds of thousands of trawl hauls, and total documented scientific specimens that likely number in the hundreds for a species distributed across the Atlantic and Indo-Pacific. For an animal of its size and apparent distribution, the encounter rate is extraordinarily sparse. Captivity provides the clearest explanation for why. Every frilled shark brought into human care has died within hours. The failure is physiological rather than logistical. These animals inhabit water at near freezing temperatures under hundreds of meters of hydrostatic pressure in total darkness. The pressure change from a 500-meter habitat to the surface represents the removal of roughly 50 atmospheres of compression from an animal whose cellular biology is calibrated for it. Barotrauma begins immediately. Temperature shock compounds the injury. Aquarium water is warm relative to the abyssal environment this animal requires. Light exposure after a lifetime in darkness adds physiological stress the animal has no mechanism to manage. Then there is the capture itself, the net haul, the handling, the containment. No documented case exists of a frilled shark successfully maintained in human care for any meaningful period, because every element of the surface world runs contrary to everything the animal's physiology was built around. The frilled shark has no such buffer. Every feature of the encounter with the human world is lethal. The Awashima footage from January 21, 2007 exists because the animal was already separating from the environment that kept it alive when Kanbe found it. Whatever drove it into shallow coastal water, illness, disorientation, injury, it had already begun the process. Park officials told Reuters they believed it came to the surface because it was sick, or else it was weakened because it was in shallow waters. Both were almost certainly true at once. Staff filmed it for a few hours, then it was gone. That 2-hour film record is what passes for an extended encounter with a living frilled shark. The footage has accumulated over 8 million views online since it was posted. The staff who described it as of incalculable value weren't exaggerating. The frilled shark's teeth are the most instructive thing about it. Every tooth in both jaws is tricuspid, 3 main pointed cusps with smaller cusplets between them, all needle-narrow and sharply recurved toward the throat. The dentition is homogeneous throughout the mouth. Teeth at the front of the jaw share the same fundamental architecture as teeth at the back. Most modern sharks carry differentiated dentition, distinct shapes for grasping near the front, cutting at the sides, and sometimes crushing toward the rear, a functional suite matched to the dietary complexity of an opportunistic predator encountering prey of varying size and texture. The frilled shark carries one design, deployed uniformly from front to back across dozens of rows in each jaw. That single design is matched precisely to its primary prey. Squid are soft-bodied, slippery, and capable of rapid directional changes. A single-point cusp can lose them. The frilled shark's 3 cusped teeth function as multiple simultaneous hooks, and the sharply recurved orientation means movement in any direction once contact is made tends to drive the cusps deeper rather than release the grip. A to Z Animals describes the teeth as needle-like and multi-cusped, helping grip slippery prey like squid during a rapid strike. Combined with the highly distensible jaw, capable of accommodating prey up to half the shark's body length, the design allows the frilled shark to retain and swallow squid of substantial size without needing to reconfigure between contact and capture. The body enables the strike the teeth complete. The elongated trunk and far posterior fin arrangement, single small dorsal fin near the tail base, broad anal fin larger than the dorsal, small paddle-shaped pectoral fins, create a morphology unlike any other living shark. The inference from body mechanics: the frilled shark compresses its elongated torso laterally before releasing in a rapid lunge, the terminal mouth closing on prey from a distance the typical shark body can't manage. No direct footage of this strike in the wild exists. The animal has never been observed hunting in its natural habitat. The behavior is inferred from anatomy, but the anatomy argues strongly, and the stomach contents confirm what the inference predicts: a diet dominated by exactly the soft, slippery prey that this particular tooth and body configuration was built to catch. Compare this to the tooth designs of the earliest sharks in the fossil record. Devonian sharks carried cladodont teeth, multi-cusped, with a large central blade and flanking cusplets, described in paleontological literature as best suited to grasping prey swallowed whole. Author John Long, in The Secret History of Sharks, describes this early multi-cusped architecture as built for exactly that function. The frilled shark's tricuspid teeth represent a refined version of the same functional logic: multi-pointed, grasping, optimized for soft prey that can't be cut into portions. The design is a precision tool for a specific ecological task, and it has been confirmed as effective by every generation of frilled sharks that successfully caught a squid and survived long enough to reproduce. This is precisely what the label living fossil tends to obscure. Charles Darwin coined the phrase in On the Origin of Species in 1859, originally describing organisms showing minimal evolutionary change. The phrase has since accumulated a connotation of stasis as failure, as if persistence without obvious revision were a kind of evolutionary embarrassment. The frilled shark inverts that logic entirely. Stabilizing selection as a mechanism does not mean nothing happened. It means every deviation from an already optimized design was removed by selection, while what remained was continuously confirmed. The frilled shark's current form is the accumulated result of roughly 80 to 95 million years of that confirmation process. Every mutation that produced a meaningfully different frilled shark, different tooth geometry, different jaw distension, different body form, was filtered by the same unchanging environment, asking the same unchanging question. What you see in the Awashima footage is what selection kept, not a primitive animal, a completed one. Haruo Kanbe didn't know any of this on the morning of January 21, 2007. He saw an eel-shaped creature with a frill around its throat, moving wrong in water too shallow for it, and called the Marine Park. Staff came, netted a 1.6-meter female, placed her in a seawater tank, filmed her. The water was wrong, the temperature was wrong, the light was wrong, the pressure was gone. Everything the surface world offers is the wrong answer to every question the frilled shark's body has learned to ask. 2 hours later, the footage was all that remained. What the Awashima staff filmed that morning was an animal that had been carrying a working design through geological time since before T-Rex walked the earth. A lineage whose fossil teeth appear in late Cretaceous rock alongside the bones of animals now studied in natural history museums. A creature whose ancestors passed through the KPG extinction while the surface world above them restructured itself entirely. Because the darkness they inhabited asked nothing different from them than it always had. And the design they carried had already answered that question correctly. Every other shark alive is a revision, a lineage that encountered a changed environment and changed in response. The great white's molecular ancestry traces back roughly 60 to 65 million years. The Hexanchiformes have been traceable in the fossil record for nearly 200 million years, but have speciated and shifted throughout their tenure. The Greenland shark is a marvel of individual time, nearly 4 centuries in a single body, but its lineage belongs to a family far younger in evolutionary terms than the frilled sharks. These are all revisions, different answers worked out at different moments in geological time to questions the environment kept changing. The frilled shark received the same question it has always received, in the same water, in the same darkness, for a span of time that reaches from before the KPG boundary to the present. The answer it had arrived at by the late Cretaceous has required no structural revision since. Teeth of the same basic architecture, a body of the same basic form, a diet drawn from the same basic category of prey, a depth in the same category of ocean. The frilled shark isn't a fossil that forgot to die. It's a design that got finished early, completed roughly 80 million years ago in a world that no longer exists above the waterline, and then preserved by the one place on Earth that changes slowest. Every other shark alive is a revision. This one is the original manuscript, still swimming, still working, still proof that in the right darkness, perfection doesn't need updates. The fisherman off Japan didn't find a monster that morning in 2007. He found something stranger than a monster. He found an animal that had already been right for 80 million years and had simply run out of reasons to stop. If you want more stories like this from the deep, subscribe.